Hearing?
Hearing, yes. So we call it auditory processing, we can call it audition, we can call it processing of hearing. Understanding the process of transduction, how the brain plays a role in hearing, and when things go wrong. To start off this process of transduction for audition, certainly we need a physical stimulus. Sound waves are going to enter our ear. We've got an outer ear, a middle ear, and an inner ear. Okay, this is your pinna. We have a pinna on each side. It funnels the sound waves inward toward the auditory canal. The first structure they're going to come in contact with is called the tympanic membrane. Once those sound waves get through the tympanic membrane, starts the middle ear. So these three bones here are called the ossicles. Collectively, we call them the ossicles. I know in biology there's different terms, hammer, anvil, stirrup. In biology, I think they do like stapes, malleus, and okay, they're middle ear structures. They're three little bones that actually move. They're three separate bones, they move. We call them the ossicles. The job of the ossicles is to amplify sound. If you're a trivia person, they are the smallest bones in the human body. After the ossicles amplify the sound, those sound waves get to the oval window. Middle ear, part of the tympanic membrane, the ossicles, and part of the oval window. The sound waves are going to move over to this snail-like looking structure called what? The cochlea, from the back of the cochlea. The auditory nerve transduction takes place. This is our focus right here. The basilar membrane would be found inside the cochlea. The organ of porti is found inside the cochlea. Is actually responsible for turning the sound waves into neural impulses. Play a really important role. These hair cells are always moving. When the sound waves are converted into neural impulses, those hair cells that are moving, they send those neural impulses to the auditory nerve. We have lots of those little hair cells sitting on our organ of porti. But it's the hair cells that take those neural impulses and send them to the auditory nerve. I'm pretty certain you all know from the auditory nerve where it's going to go. Eventually it gets to the thalamus, eventually it gets to the temporal lobe. From the auditory nerve, this information is going to stop at the cochlear nucleus of the medulla, so a couple of different places. The superior olive helps us with localization of sound. Some of it, of course, is going to go to the inferior colliculus. That's of the midbrain, if you recall. That's more reflexive. So I'm going to say orients us to sound. From there, now we finally get to the thalamus. Medial geniculate nucleus. Finally, to the temporal lobe, primary auditory cortex. The primary auditory cortex is also known as Heschel's gyrus. So, in many areas, could hearing be impaired? Many, many areas. Where does transduction take place? Organ of porti, which is located in the cochlea. Let's talk about hearing impairments. So mixed hearing loss means both things. It means conduction loss. Conduction loss usually pertains to the middle ear. The ossicle's not formed correctly, or there's too much fluid and the bones can't move and amplify as correct, can be helped with hearing aids. Hearing aids are little microphones and they amplify sound. They just make things in the environment louder. People who have hearing aids, everything is amplified. So their brains don't screen out what's irrelevant. You have to use whatever energy and effort you have. Try to focus in on just paying attention. So our vestibular sense has to do with the tilting and the movement of our head. That ultimately plays a huge role in our sense of balance. It's made up of two main parts. Otolith organs, which are the utricle and saccule. And if we took a little cross-section, we would see that gel layer. These little things on top, they're like tiny crystals, are called otoliths. When our head is straight, those little tiny crystals or stones are just kind of sitting on top of that gel-like layer. If we were to tilt our head back, those little stones are going to move. And when they move, there's hair cells again. And so that is going to detect the movements. And that is going to give information to our somatosensory cortex. The cranial nerve, the auditory or cranial nerve 8, responsible for taking the information from the otolith organs to the somatosensory cortex. The semicircular canals, also part of the vestibular system. 3 chambers filled with fluid. Movement of the fluid within those 3 canals. From the auditory nerve, the information is further processed by the vestibular nuclei in the pons, medulla, and cerebellum. These 2 structures make up the vestibular system. These have the little stones and the gel-like substance inside. The semicircular neurons have fluid inside. Any movement of the stones or of the fluid is picked up by the auditory nerve. That information is sent to the vestibular nuclei located in the pons, medulla, and cerebellum. Some of this information is sent to our spinal cord to adjust our posture. Systems in place that help us keep our balance. This is one of them. Vision also helps us keep balance, so you can see where you are. Your cerebellum also helps you keep balance. It has to do with muscle tone. This information eventually is going to get to the somatosensory cortex. The ventral posterior nucleus of the thalamus is the particular area of the thalamus that's responsible for our vestibular sense. Some of it goes to the spinal cord so we can adjust our posture. Some of it goes to this part of the thalamus. Finally, somatosensory cortex. Okay, so this is a much shorter pathway. Your skin. It is the largest organ we have. Sensory receptors, mechanoreceptors, are embedded within our skin. They're going to travel through our spinal cord, a thing called the dorsal column medial lemniscal pathway. Skip right over to where it goes next. Part of the thalamus is involved. It is the ventral posterior nucleus. The big thing is knowing the sensory receptors, knowing the process, knowing the part of the thalamus that's involved, and knowing the part of the cerebral cortex that's involved. Here it's the parietal lobe, specifically the somatosensory cortex. This is we don't need to write this down. It's called a pathognomonic sign. It means it's something very unusual that we see, that we know right away what's wrong. They're missing the left side. Is what we call neglect syndrome. Neglect syndrome is very easy to detect when we have people draw what they see, because they will ignore stimuli on the left side of the visual field. Their actual vision is fine. They ignore the left side, yet the interesting thing is when we say, take a look over here, take a look at yours, they'll go, oh my gosh, look at that, I forgot the petals. And then they realize, but unless their attention is directed to it, they will completely ignore the left visual field. Called neglect syndrome, caused by damage to the right secondary somatosensory cortex. When the right secondary somatosensory cortex is damaged, it results in impairments in visual perception. Not vision, visual perception. It has to do with what they see in space. They will not do it on their own. So this is called neglect syndrome, sometimes called unilateral neglect syndrome. Same thing. All right, so we're done with the vestibular sense. We're done with touch. The receptors for temperature, where would they be located, by the way? In your skin. We also, embedded within our skin, have what we call thermoreceptors, are for temperature detection. And then olfaction and gustation are going to be on your own. Do you guys know what olfaction is? It's a fancy name for what? Smell. Sense of smell. How about gustation?
Digestion.
Taste. Right, these are the receptors that respond to pain. Okay, so either pain we call mechanical manipulation or pain caused to high or low temperature. Nociceptors pick up on both types of pain. Chemicals can also cause pain. Have you ever eaten hot peppers? Worked out really hard, and the next day you pretty much can't move. Hurts so bad. Lactic acid can activate nociceptors. So there's a couple of different ways that these nociceptors can be activated. The pathway for pain is that these nociceptors are activated by a variety of means. Travel through the spinothalamic pathway, synapse with a particular part of your thalamus. For pain, it's what we call your intralaminar nucleus. The thalamus is going to send this information to one or two or both places. One of them is somatosensory cortex. So the primary somatosensory cortex. So that's generally physical pain. Heartbreak or emotional pain. All right, so if something really horrible happens, sometimes people, they get a sensation of physical pain. But it's your anterior cingulate cortex can also be activated. The ACC, anterior cingulate cortex, part of the limbic system, that is activated in emotional pain. You get your heart broken, certain chemicals that are released in your brain after getting rejected are going to activate your nociceptors.